Behavior may be the product of a brain structure. Means behavior and brain structure are connected to each other. There is a deep connection between these two. So what is this connection? According to localization states that each part of the brain, there is a certain area in the brain that is linked to psychology. For example, maybe this part of the brain is for fear, this particularly area is for memory, this particular area is for anger, this is for your personality, this is for your temperament, your hope, your faith. So there are, for every particular behavior, for every specific behavior, there is a specific region in the brain that is connected to it. So before we actually understand what localization is, just get familiar with the basics of the brain here. Let's understand the structure of the brain and we proceed forward. Our nervous system is a bunch of neurons that are connected with each other, and these are the neurons which are responsible for any communication from the outer world or inner communication that is linked with your brain and spinal cord. This central nervous system includes your brain and the spinal cord. So if you see, this is a human brain, a model of a human brain. If you see, there is this deep furrow between the left and the right hemisphere. So this is left hemisphere and this is right hemisphere. This deep furrow, here there are neurons that connects the left hemisphere to the right hemisphere. So neurons are here connecting both the hemispheres. These neurons are called as corpus callosum. Talking about the lobes, this is your frontal lobe, this is your parietal lobe. At the back, it's the occipital lobe, and on the sides are the temporal lobes. So this part that you see, this is called as thalamus. Thalamus is where the sensory nerves from all over the body brings in the information, all the communication, and it goes here to the thalamus. So this is the hub of communication. From here, it then further goes to the particular parts of the cortex. Then down below is the hypothalamus. It is just below the thalamus. It is responsible for your hunger, thirst, and basic things. Right here is amygdala. Amygdala is very, very important in psychology because it is related to your memory as well as emotions. So when the emotions are triggered, those things get feeded in the memory, so that amygdala decides how intense the emotions are, and that particular memory is to be stored or not to be stored, or where it is to be stored. So amygdala is very important when we are studying memory and emotions together. So we'll be talking about amygdala later on as well as we study memory. And hippocampus is your important part of the brain which helps in learning, memory, and it also transfers your memory or information of the memory from short-term memory to the long-term memory. So if it is from the hippocampus that the memory gets transferred from STM to LTM. And then we have a brainstem down here which is basically responsible for all basic kind of functioning, your heartbeat, your breathing. This is there in lower animals as well. So this cortex is basically the gray area, the intelligent part which is there in humans, more in humans, and very less in the animals, because this is the area. We'll be talking about it later though, about the intelligence, but right now only the brainstem we are talking about, that is definitely there in the lower animals. So now talking about localization, initially the researchers stated that a particular damage to a certain part of the brain actually leads to malfunction of that certain behavior. Like for example, just an example, if this part is responsible for your memory, then the person is going to have problems in memory if this part gets damaged. If this part is for learning, and if this due to any injury or accident a person gets hurt, or the brain gets damaged, only this part, then the person is going to have a problem in learning. So specifically whatever malfunctioning is happening, it is because of that particular area of that brain has got damaged, or there are some kind of lesions there. You have understood what localization is, that every behavior has a certain area related to a certain part of the brain. Now, to support this, we always have to learn some studies in IB psychology. So here I'm going to support the study by a very famous study, Paul Broca. Study's name is TAN. It is by Paul Broca in 1861. It's quite an old one, but a very interesting one. So in this study, there was a participant. It is a case study. So that means that only one participant is there that we are going to talk about. Louis Leblanc was only at the age of 30. He was later known as TAN. Okay, so Louis Leblanc was later known as TAN. So TAN was actually developed some gangrene, and due to which he went through the surgical procedures where he lost the function of speech. After this problem, the person TAN or Louis, he was only able to speak the word TAN. That is it. So now talking about this case, now TAN only had the problem of speech impairment was there. He could only speak the word TAN, and rest of the functions were absolutely fine. Unfortunately, only when he died, the researchers could do the post-mortem and see what exactly the problem was. They saw that there was a certain area in the brain which is related to speech that had got damaged. So due to which he had a speech impairment. Now this person had all the other functions going pretty well. His personality was absolutely fine. He had no problem in his intelligence level. He was able to understand what other people used to talk about. He was able to comprehend the language as well. But when it came to speaking, only then he could speak the word tan. For what reason, that we don't know, but he could only speak those words and nothing else at all. So that is why he was known as TAN after this study. So during the post-mortem, they saw that it was only particular, very specific part of the brain that had gone damaged, due to which he had a speech impairment in that he could not speak. So here the study clearly supports the localization, that one particular behavior is being affected when one specific part of the brain gets damaged or gets affected in any way. So Paul Broca was the person who did the research on this person, and so this is called Broca study, and this specific area of the brain is known as Broca's aphasia. So Broca's aphasia means the problem in the articulating of the speech. So not just TAN, but he also did research on 25 other patients, and only then he came up with this result, with his findings, that there was this specific part, that is the left hemisphere, frontal left hemisphere, is particularly related with the known as Broca's area. So after this study, this researcher gave the name of this part of the brain as Broca's area, which is related to the articulation of speech when the person is having a problem, that is known as Broca's aphasia. Later on, further mapping was done on the same, and then the researchers found out another area called as Wernicke's area. In Wernicke's area, the person is very clearly able to speak and they do not have any problem in speech, but they have a problem in the comprehension of the language. Wernicke's area is particularly located in the temporal lobe of the dominant area of the brain. Dominant area is usually the left hemisphere of most of the people. So it is responsible for the comprehension of written and spoken language. So when a person is having Wernicke's aphasia, if this part of the brain specifically gets damaged or injured, the person has got Wernicke's aphasia. In that case, the person is able to speak very fluently and very naturally. They do not have any problem in their speech, but the only thing is they are not able to comprehend the language. They are not able to understand and make meaningful speech. So in that case, though they're speaking normal, the words that they're articulating, the speech, that is absolutely normal, but it is completely meaningless because they're not able to form the meaning out of the sentences that they are speaking. So let's do one study that supports localization. One of the very famous study, H.M. Milner, 1966. We call it H.M. As a short form. So H.M., when he was just 7 years old, he was staying in Connecticut and he got hit by a cyclist when he got very badly injured and he got severe head injuries. After having a serious head injuries, he started to having epileptic attacks at the age of 10, and it was assumed that it is connected with the accident. So by age of 27, he started having so many epileptic attacks on daily basis that he could not lead anymore a normal life, and he was suffering very badly. There was no cure at all. Even the medicines that he was having could not cure him. So after that, one of his doctors, a neurosurgeon, William Conville, he decided to do an experimental study on him. So what he did, he removed a tissue from HM, or from the temporal lobes, and some tissue from the hippocampus. This surgery was done. He cured from the epileptic attack, so it was a successful surgery. But after many years, Brenda Milner, who is the researcher of which the study we are talking about, Milner came to meet HM, and she realized that HM would forget daily events so much. He became so forgetful. Would forget things as soon as they would occur. He would meet people and forget their names. He would do some task and he would forget that he has done that task, or somebody would give instructions to him. He could not fulfill those instructions and he would just forget. But after the operation was done, so Brenda Milner, she studied HM and came up with some findings that HM remembered everything before the operation, before the surgery that had happened. So he remembered his childhood very clearly, but after the surgery he had a lot of forgetful events and he could not remember most of the things. His personality was unchanged. There was no change in his personality as such. Even there was no problem with his intellectuality. He had no intellectual impairment. He was absolutely fine with that as well. So he was in particularly suffering from retrograde amnesia. Retrograde amnesia means he remembered things, or he had memory before the operation. So whatever happened before the operation, he had memory of that, but after the operation, after the surgery, he did not have any memory, almost no memory at all. So that is called as retrograde amnesia. After a while, he recovered from retrograde amnesia and he started to suffer from anterograde amnesia. Unable to remember the faces of the people he had met in the morning. By afternoon he would forget, and he would behave in a way that these people who have come to meet him, their psychologist or the doctor, they are very new people and that he had never met them before ever. Procedure and results of the study. Now the aim of the study was to better understand the effects what the surgery had on the patient HM. So in order to carry out, there were different strategies used. Like I said, it was a case study. Case study, as you all know, has usually a triangulation method. Means we have a lot of different kind of research collection data methods. There could be observation, interviews, then questionnaires and so on. So in his study, there was psychometric testing was done. This was to see the IQ test of the HM, and the results were above average. That clearly shows that he did not have any kind of intellectual problem at all. Then direct observation of his behavior was done. Then interviews were held with HM and his family members as well. There was also cognitive testing done on the memory recall test as well as learning tasks such as reverse mirror drawing. So now later, Corkin did an MRI to determine the extent to which the damage had been done to HM's brain. And HM could not acquire any new episodic knowledge, that is memory for events, and he could not acquire new semantic knowledge, that is general knowledge about the world. This suggests that the brain structures that were removed from his brain are important for the transfer of information from short-term to long-term memory. So that means it clearly shows that he had no intellectual problem, he did not have any personality problem, he did not have any other all functional problems. That means all his brain, after the MRI, it clearly showed that all the parts of his brain were perfectly well intact, were in place. But there was this specific part that was removed from his brain, was the only part responsible for transferring the memories from short term to long term. So that is what he was actually suffering from. He would meet people, he would do tasks, but he could not remember that after a few minutes or hours. Why? Because whatever he was doing or whomsoever he was meeting, those things were getting registered in his short term memory. So from short term memory, he was unable to transfer that information to the long term memory. So that means those tissues that were removed from hippocampus and the temporal lobes, those were responsible for transferring information from STM to LTM. So the researchers also found that he was able to remember his own house, and he could draw a picture of the floor plan of his new home. So this indicates that cognitive map of the spatial layout of his house, that was absolutely intact. There was no problem in it. So that means a memory is not encoded in the same way as semantic or episodic. Now this is very interesting. The person is not able to remember the episodes. The person is not able to remember the meaning of something semantic like knowledge or any kind of facts. They are not able to remember. But they are able to remember the ways of their house, the map of their house, the blueprint of their house and everything. Why? That clearly shows that memory is not just specific to one particular thing. Everything is not stored at one place. Semantic memory is different from procedural memory, and spatial memory is altogether different from episodic memory. So there are different storages for all these things, and everything does not get stored at one place. Now HM had a capacity of working memory since he was able to carry on normal conversation. This requires minimal level of retention of what he would just hear and say. So after on being asked to recall the number 584, HM was able to do so even after 15 minutes, but only if he had constant rehearsal of motor skills, that is procedural memories, where it is well maintained. For example, he knew how to mow a lawn or do tasks like mirror drawing, in which he had to acquire new eye-hand coordination. Although he showed improvement on the skill over time, but he never remembered learning the skill. So every time Miller asked him to do it, he would say that he's never tried before. So Corkin carried out another MRI scan of HM brain to see that extent of the damage. It was possible to see the parts of HM's temporal lobe, including hippocampus, had the most damage. So hippocampus was the part that was the most damaged, and this clearly shows the localization that specifically is responsible for transferring the memories from your STM to the LTM. This is the summary. The memory system in the brain is highly specialized and a complex system. Hippocampus plays a critical role in converting memories of experience from short-term to long-term memory. Well, the researchers found that short-term memory is not stored in the hippocampus because the hippocampus was removed from HM's brain. That means short-term memory is not stored in hippocampus, it is stored somewhere else. Hippocampus is only transferring the memory because HM was able to, recall or have a, within the 15 minutes he would able to recall whatever was said to him, or he was able to carry a normal conversation. So since HM was able to retain some memories for events that happened long before his surgery, it indicated that the medial temporal region is not the site of permanent storage, but it just plays a role in the organization and permanent storage of memories elsewhere in the brain. So that let's do the evaluation of this study. The first thing is that it was a case study and it was a longitudinal study. It went over for 50 long years. So the good part is, the strength of this study is that it has a lot of data, in-depth, rich data collected by the researcher. So having a rich quality, in-depth data is a good strength of the study. But the weakness of the same would be that a person, a researcher can get very close to the patient. They become close and it can lead to researcher biasness. So your perception changes, your emotions come in and you can become a little biased. And another strength of the study was method triangulation. So there were a number of research methods used for it: observation, interviews, survey, psychometric tests, and so on. The limitation of this case study was that it cannot be replicated. It's a very unique case and there was just one participant for it, so it cannot be replicated, and it is difficult to maybe generalize the same. Some of the study was retrospective in nature. This means that we do not have a lot of data on HM, actual cognitive abilities before the accident. High ecological validity, everything was conducted in his very natural environment, so this study is of very high ecological validity. Then it also met another good thing is that it meets all ethical guidelines: privacy, confidentiality, protection from harm, and so on. Have you ever sit in a taxi? And have you ever seen that taxi drivers, when you are telling them to go by the main road, and if there is too much of traffic there, they just, turn their wheel and steering wheel, and they can take the inside roads, the peripheral lanes, and they can still get you out of that traffic jam. So how do they really know all this map of the city? It's like on the back of their hand, isn't it? Their spatial ability is amazingly well. So we want to know biologically, is the brain of a taxi driver any different from the brains of other normal people? So what is the biological difference here? So this study is of Maguire 2000, a very interesting study. This was to see that the London taxi driver's brain was any different from the other common people's brain. And in this study, there were around 16 right-handed males London taxi drivers, and they were all compared with the MRI scans of 50 right-handed males who did not drive taxis. So there are two experimental conditions. So there are two conditions here. One is experimental group, in where we have 15 taxi drivers, all are right-handed males, and on the other hand, in the control group, we have non-taxi drivers, which are 50 males. And in order to take part in the study, the participants had to give the knowledge test and a license that would say that, they had license for over 1.5 years minimum, and the MRI database was collected. So the sample included a range of ages, so there was no confounding variable at all. The study is correlational in nature because there was no IV as such manipulated by the researcher, and the researcher was looking if there is a relationship between the number of years of driving taxi and the anatomy of one's brain. So it was a single blind study that the researcher did not know whether she was looking at the scan of a driver or a control. It was a single blind study. The researcher did not know whether she is looking at the MRI scan of the driver or of a non-driver. So that was a single blind study to remove any research biasness. The data from the MRI was measured using two different techniques: voxel-based morphometry, that is VBM, and pixel counting. So what is voxel-based morphology? It is used to study to measure the density of gray matter in the brain, and pixel counting consists the counting of the pixel in the images provided by MRI scans in order to calculate the area of the hippocampus. So one is the gray area of your brain, and the other is the hippocampus, the area of the hippocampus that was studied in this study. Now there are two main key findings of the study. So the pixel counting here showed in an MRI that the posterior part of hippocampus was larger in taxi drivers than the non-taxi drivers, and the anterior part of hippocampus was less in the taxi drivers as compared to the non-taxi drivers. So what exactly does this mean? And the VBM showed that the volume of right posterior hippocampus correlated with the amount of time spent as a taxi driver. So the volume of right posterior hippocampus was larger, or was correlated with since how long they have been into driving the taxi. So what exactly does it mean? It means that posterior hippocampus is involved when previously learned spatial information is used, whereas the anterior hippocampal region may be more involved during the encoding of new environmental layouts. let's do the evaluation of this study, and this clearly shows that there is definitely the difference between the brain structure of a taxi driver if you compare it with a non-taxi driver. So the evaluation of the study was, it was a quasi-experiment. That means there is no cause and no effect in the relation, no effect, cause and effect relationship can be established. So there is no independent or dependent variable, it was naturally occurring. The brain scans were coded so that analysis could be done blindly. The researchers did not know that they were looking at the MRI scans of whom. Is it the driver or the non-driver? So it avoids any kind of researcher biasness. Since this is a correlational study, it is difficult to say what is the cause and what is the effect. Is it that because they were taxi drivers, that is why their posterior hippocampus was large? Or it could be the other way around. Because the posterior hippocampus was large, they decided to become a taxi driver. Like for example, if you have a good skill in something, if my posterior hippocampus is big and I'm very good in understanding the spatial ability, I'm very high in spatial ability, I understand the road maps and everything very quickly, maybe I decide to become a taxi driver because I'm good at it. Then another is that it appears to have a sampling bias because all were from London, all were right-handed, all were males. So this is a kind of a biased sample that we have in the study. There are no females, there are no non-London, there are no left-handed drivers in this. That means this study cannot be generalized to a larger population but to a very specific kind of a population. And ethically sound it is because MRI is done, and it does MRI, any kind of biological test always gives a very objective result and very trustworthy, reliable result. It is not subjective at all. So MRI was done and it is not at all harmful. So it is a very good ethically used test. So these are the 2 to 3 studies that you can write in your exam when you are writing for localization, that is in favor of localization. Now I will be telling you one study that you can write as a counter study in your localization, where the study is against localization. If the question comes, then you can use the study. So Carl Lashley in 1890-1958 he used the technique of measuring behavior before and after specially carefully controlled induced brain damage in the cortex of rats. Carl Lashley gave a study where he studied that memory is not localized. So what he did, he took rats and he had put them in the maze. So rats over a period of time gradually had learned the way to the maze. So they would know where to, find the food in search of food. They started knowing, understanding and memorizing the ways through the maze. So after what he would do, gradually little by little, he kept removing a part of their cortex. So when he kept removing the part of their cortex, the rat would still remember their way through the maze. If memory was localized, if there was a specific part where the memory is located in the brain, in that case, at one point of time, Carl Lashley would have succeeded in a way that when he was removing cortex one by one, at one point of time, the rat would have actually forgotten the way of the maze, because that particular part that was responsible for the memory is gone out of his brain. So, but this did not happen. So this clearly shows that memory is not localized, it is lateralized. It is all spread throughout the brain, and it is not any specific area, it is not localized. So there is this principle of mass action that says there is a correlation between the percentage of cortex removed and learning ability. So lesser is the cortex, slower and more inefficient is the learning. So people who have more cortex, more gray area, they are likely to be more intelligent and more efficient learners. Second is equipotentiality, that is the ability of one part of the cortex to take over the functions of another part of the cortex. So that means if this is your brain and one part of the brain gets damaged, or there is some lesion or something has happened, it gets injured. So if that specific part of the brain has got damaged, so that means you have got any dysfunctionality in one of your behaviors. But that is not the case. It can, the other parts, the nearby parts of that cortex can take over the work from that particular part that got damaged and make over the functions. It's just like you have a company, you have an organization, and I'm the boss, and I have one HR team, one finance team, accounts team, R&D team, production team, and so on. So these are all my teams in the organization. If one person is absent, is having an off that day, so that does not mean that work will not be done. The other people around that person who know that, this is a kind of a role, or this is a kind of a thing that he does, the person who is on off, so they take over and start doing his work, and my work keeps going on. Similarly, brain is very smart and works in a very similar manner. If a particular part of the brain gets damaged, or there is some injury that has happened in a particular part of that brain, so what happens is the nearby parts of the brain, they take over the control and become more sharp in either other areas, and to some extent can take over the functions of that damaged part of the brain. So we'll be covering on to that more later in the chapter. For now, there is another study, relativity of localization, the split brain research. So in this study by Roger Sperry and Michael Gazangia in 1967, they gave a study in favor of lateralization, saying that, your brain works as a whole and the functions are all lateralized, are all spread out, and it's not just only the specific part of the brain that is responsible for one specific behavior. So things are quite, spread out in the brain. So he did a study, and one of the theory was tested, where in the beginning of the class, I told you that this is the corpus callosum, the neurons that connect the left hemisphere with the right hemisphere. So people who have epilepsy, who suffer from seizures, if this corpus callosum is dissected, if this corpus callosum is removed, if this corpus callosum is cut, the person gets relief from seizures. And there was a study in which the experiment in which patients actually who were suffering from severe epilepsy got their corpus callosum cut, and they realized that they had actually got cured from epilepsy, and they did not find any difficulty or any problem at all in their normal functioning. So there was no difficulty in their normal functioning, and they were not suffering from, they were cured from the epilepsy. So what could be better than that? So on to that, there was this study given by the Sperry. So now in this study, if you see, there is a picture of spoon that is left visual field, right hemisphere. So before we actually start with the study, I would just like to tell you, if you know already, then fine. The ones who do not know, your right side of the brain takes care of the left side of the body, and your left hemisphere takes care of the right side of the body. So it's like cross connected. Okay. So now in this, a picture of a spoon is there on the left visual field, that is in the right hemisphere is being seen. So there is a spoon here, and basically it is the right hemisphere that is activated right now and seeing the spoon. So when asked, patient said nothing. When asked to pick, patient felt and picked spoon kept behind the screen. Now patient could not explain why he picked the spoon. Patient used right hemisphere and used left hand to pick. So center of speech is in left hemisphere. So now in this study, what was happening, that participants were sitting, and in front of the participants there was a screen, and in this participants the corpus callosum was cut to cure their epilepsy. They did not feel any difference at all, but the researcher wanted to study that there is a lateralization of the language. So what he did, behind that screen there were some things that were kept. The person could not see, only the hands would go. Now the participant was shown a picture of a spoon. So picture of a spoon was shown to the person, and then he was asked to pick it up. So his right hemisphere is working. He saw the picture of the spoon in the right visual from the right hemisphere, and from the left hand he just felt and picked up the spoon. So that means his right hemisphere is working, he saw and he picked up from the left hand. When he was asked that why did he pick up the spoon, he could not say anything. There was no explanation from the participant side, because the articulation of speech, the expression is in the left hemisphere. So that is why he could not. And the corpus callosum here was cut. So when it was cut, there was no connection or no communication between the left and the right hemisphere. So the right hemisphere saw the spoon, picked it up, but the left hemisphere, he could not express that why he picked up the spoon, because the communication is not happening between both the things. So this clearly shows that language was comprehended by the right hemisphere, but the language could not be spoken because it is the left hemisphere that is responsible for the language. So this is the proof of lateralization. Language is spread out and it is not any particular part of the brain that is responsible for language. So we are done with the localization and lateralization. The question would come on localization, not on lateralization for IB psychology students. So now if you have any queries or anything that you have not understood or want more clarification on the same, please write in the comments below and subscribe to my channel to learn more lessons on IB psychology cognitive approach.